Good afternoon. Welcome to Medical Grand Rounds. I'm Bob Wachter. I'm chair of the Department of Medicine. Today, you see the title up there, Our Emerging Understanding of the Gut Microbiome in Health and Disease. Really looking forward to this. Just a program note that next week we had planned to do a clinical problem solving session, but have decided that there's enough going on in COVID and Omicron that we'll do a Omicron session, still putting it together, but you can assume it'll be terrific speakers and state of the art about what's going on as our understanding changes about every 5 minutes. So that'll be for next week. For this week, thrilled to be doing this session on the microbiome, potentially one of the most important developments over the past decade or two. The ground rules for the session are here. I think you know all these right now. We'll try to save some time for Q&A at the end. If you have questions, type them in the Q&A box. This, like all of our grand rounds, will be posted later on our website and on YouTube, probably end of the week or beginning of early next week. And closed captioning is available to you if you would like. So this is an incredibly exciting, emerging area that we are lucky at UCSF to be really national and world leaders in our understanding of the microbiome, both from a kind of foundational basic science all the way up to the clinical and translational aspects of it. Much of this work is centered in the Benioff Center for Microbiome Medicine. And so today we're going to have a tag team that takes us from bench to bedside, helping us understand the latest and greatest in this emerging and really important area. Our two speakers in order will be first Sue Lynch, who's director of the Benioff Center for Microbiome Medicine, also professor of medicine in our division of gastroenterology here at UCSF. She's a graduate of University College Dublin and did her postdoc at Stanford and really has staked out a role as being someone who truly understands what's going on with the microbiome and is advancing our understanding as it pertains to predicting, preventing, and treating disease. So look forward to hearing from Sue. And after Sue, we will hear from Najwa El-Nachef. Najwa is associate professor of medicine, also based in the GI division in our department. She runs our spectacular GI fellowship, which just had a fabulous match. She's a graduate of that fellowship herself after completing her residency at the University of Michigan. She's an active clinician, and her translational research interests are particularly focused in inflammatory bowel disease, and you'll hear today about some of the exciting work that's being done in IBD as it relates to the microbiome. So with that, let me hand it off to Sue to start, and we'll get to Najwa afterwards. Thank you both for being here.
Great. Thank you so much, Bob. And I'm absolutely delighted and thrilled to be with you today to share some of the history of the field of human microbiome research, where the field has evolved over the last 15 years or so since its inception, and where we're going in terms of thinking about how we translate the observations and findings that we're making in human populations into new therapies and treatments for a variety of diseases. And as you heard, I'll pass the baton over about halfway to Najwa, who'll tell us more and dig in deeper into one aspect of leveraging microbiome manipulation in the gut for the treatment of ulcerative colitis. Disclosure, I'm a director and consultant for Shields Therapeutics. I'll talk a little bit about a trial that's going on to prevent and allergic asthma. But let's start. It really is based on observations that were published in the mid-1980s in the field of environmental microbial ecology, where James Staley and Alan Konopka observed that the diversity of microbes that they could see under the microscope from terrestrial or aquatic ecosystems was never recapitulated without the need for ever growing them in the lab. And the workhorse of the field, and what was developed back in the late 1980s by luminaries like Norman Pace in the field of environmental microbial research, were things like amplifying genes like 16S from bacteria, or ITS2, the interspacer region between ribosomes and fungi, and examining the sequence of those amplified products. And the variants in the sequence at those sites or in those genes gives you a sense of which microbes are present in a mixed species community. And so this was the workhorse for a number of years. And really, we were a little late to the game in the field of human microbiology. We really only started adapting these tools to examine human communities of microbes about 15 years ago. But because we've recognized that humans are a site for diverse colonization by microbes, we've seen a very rapid expansion and evolution of tools in the field to examine human microbiomes. The use of next generation sequencing to examine not just genes or regions of microbes for their identification, but rather sequencing whole genomes of mixed species microbes through shotgun metagenomics has really expanded our idea of the functional capacity of microbes in the human system. Using RNA, we've used RNA-seq approaches to understand the transcriptional activities of these mixed species microbiomes. And advances in mass spectrometry are allowing us to examine whole suites of proteins and small molecules or metabolites that are being produced by these microbes in situ to understand not just who's there, but rather their functional capacity, what they're transcribing and what they're producing as they interact with the human host. Coupled with these next-gen analytical tools is the critical use of model systems, everything from germ-free mice to primary immune cells and model cell lines for deconstructing the observations made in these big data analyses to understand mechanisms that underlie interactions between microbes and their hosts. And that's really elevated the field and led us to some really critical new findings and understandings of how the microbiome shapes human health, some of which I'll discuss in the next few slides. So what have we learned through the application of these tools? Well, we realized that we're actually superorganisms. We are colonized inside and out by a huge diversity of microbes. And this clear phylogenetic distributions and community states that we see in specific body niches that repeat across individuals. What we also note, and you can see here on the skin, is that this is quite distinct at different body sites. This is just simply looking at the diversity of microbes that are found at various sites across the skin surface. What's also apparent is with mass spectrometry tools, if we examine the biochemical diversity of microbial-derived molecules, these are molecules that are not produced by the human genome, at these sites, we can see even though there's very few microbes that are found, the biochemical diversity that's present at these sites can be quite large. And this is not just exclusive to the skin. We now know that there's spatial organization of microbiomes along the length of the gastrointestinal tract and in the respiratory tract, for example, as well. So this is clearly a consistency in the types of microbes that we find at these sites, at least at the phylum level, these higher order levels of classification of microbes. But it's not just spatial gradient that microbes exist on in the human host. They also evolve over temporal scales as well. All of us are born with a very simple microbiome that we can see actually in meconium, suggesting that it may be laid down in utero, and we'll talk a little bit about that later on. And we rapidly accumulate microbes over the first several years of life. By age 3, the phylogenetic distribution of microbes in the gut, and we're now starting to see this is true elsewhere at other sites like the respiratory tract, is fairly stable and resembles that of an adult phylogenetic distribution of microbes. But the functions in that very early life microbiome are quite different from that of an adult. The microbiome continues to evolve over time. It's influenced by a whole range of exposures, as you might expect. And some of these, I just want to point out here, really are key selective pressures on the microbiome. Pharmaceuticals and antimicrobials, as we now know, can have profound effects on the types and activities of microbes found in the human microbiome, but also microbes in the system can transform those pharmaceuticals and antimicrobials into more toxic states. They can deactivate them. And actually some wonderful work from Peter Turnbau's lab has demonstrated that the bioavailability of the cardioglycoside drug digoxin is dependent on whether the gut microbiome houses a strain of Egertella lentha that has an operon that reduces the digoxin to its inactive form. Peter's lab has also had some beautiful work showing the effect of diet on the microbiome. It's a very strong selective pressure, as you might imagine. But also what's really key is that in adult healthy individuals, a dietary intervention, while it causes a short-term impact on the microbiome, that microbiome shows inherent resilience. It rebounds after the dietary intervention is removed, suggesting that there's strong internal and intrinsic selective pressures on microbiomes once they've established to retain a specific composition and state. And this is also true not just of commensal healthy associated microbiomes, but it's also been true in the case of obesogenic microbiomes where dietary interventions have short-term impacts, but then the microbiome rebounds and returns to an obesogenic state following the removal of the dietary influence. So really starting to give us a sense of why these interventions may work for shorter periods of time, and perhaps understanding the resilience of these microbiomes, pathogenic and healthy associated microbiomes in individuals. But this resilience has made us, in the microbiome, has really made us think about early life exposures and early life seeding of the microbiome and how specific organisms that come into the system in early life may actually sustain and be persistent in these ecosystems over time. And we're particularly interested in early life in our lab and many other groups because this is a critical period, obviously, of microbial development that parallels critical development in the immune system and physiological development. And what we know from germ-free mice is that physiological and immune maturation does not occur appropriately in the absence of microbes. So we are reliant on our co-evolved microbes for appropriate immune and physiological development. And so if this is the case, we should be able to look in very early life to predict disease development in childhood. And in fact, there's a growing body of evidence that this is truly the case. Work from our own lab demonstrated that in a cohort of 120 one-month-old infants, that there were 3 distinct gut microbiota states in those infants, and that one of them was associated with a significantly increased risk of developing allergies and asthma years later in childhood. Beyond the fact that this was a perturbed microbiome, what we showed was that it was metabolically perturbed and disturbed also, and that the cell-free products of that perturbed gut microbiome in these one-month-old infants could actually induce the cardinal features of allergic inflammation, increased Th2 cells and IL-4 production, decreased T regulatory cells and IL-10 production in vitro using primary immune cell assays. We followed up this work by following up on an oxylipin produced in high concentrations in this high-risk gut microbiome called 12,13-dihome. And an incredible graduate student in my group, Sophia Levin, demonstrated that not only does this dihome produce the effect on regulatory T cells in vitro in that increasing concentrations reduce the frequency and the IL-10 productivity of these regulatory T cells, but that this also occurs in vivo in a mouse model of airway allergic inflammation. She went on to show that this lipid is produced by specific microbes in the infant gut. An introduction of the capacity to produce this lipid into the gut microbiome exacerbated airway responses to allergy, really moving us from observations in human subjects to a mechanistic understanding as to how early life gut microbiome activities can actually promote immune dysfunction, resulting in allergy and asthma development years later in childhood. And these observations have been validated in several other large infant cohorts and birth cohorts in Canada and Europe. Beyond this, this seems to be more of a framework that's not exclusive to allergy and asthma, but more recent data has demonstrated that the same framework applies to type 1 diabetes development in childhood. A pair of Nature papers that were published in 2018 demonstrated early life taxonomic disruption in the gut microbiome associated with type 1 diabetes development in later childhood. and followed up by showing that it was depletion of short-chain fatty acid biosynthesis. This is a microbial-derived short-chain fatty acids, which are anti-proliferative, anti-inflammatory, and a fuel source for colonocytes, that the depletion of these short-chain fatty acids and this microbial activity in very early life was associated with development of type 1 diabetes in later childhood. More recently, a very similar finding in childhood development of celiac disease has also been published in PNAS. So it seems that examining very early life microbiomes allows us to predict disease development in later childhood, and we're beginning to take away at which kinds of mechanisms are priority mechanisms by which microbes may drive development of disease in later life. And this is not exclusive to the gut. In the airways, we also see very similar capacity to predict disease development based on the types of pathogenic microbiome colonizations that are evident in children in very early stages of life, in the first months, in fact. Those that are colonized by microbiota dominated by Streptococcus, Moraxella, or Haemophilus are more likely to have viral respiratory infection that results in lower febrile respiratory illness and go on to develop allergic sensitization and chronic wheeze. And the concept here is that pathogenic colonization in very early life leads to airway epithelial destruction and to chronic inflammation that is diagnosed as asthma years later. Wonderful work from Marina Sirota at the Computational Health Sciences has also shown that this is true in the vaginal tract. So Marina and her group consolidated 60 nest data from a large number of studies of the vaginal tract from mothers during pregnancy. And using a number of predictive algorithms, what she showed very clearly was that there was a very distinct microbial predictive capacity for the vaginal microbiota for preterm birth. More specifically, her team demonstrated that a depletion of lactobacillus in the vaginal tract in pregnancy was highly predictive of preterm delivery in those pregnancies. And the vaginal microbiota is unique amongst microbiotas in that it is highly dominated, typically in healthy women and in healthy pregnancies, by lactobacillus species. So the concept here is that loss of these protective species may drive preterm outcomes with the mechanism as yet to be defined. But the fact that these very early life prenatal colonization patterns in the mother matter for later later childhood development and outcomes is to some extent perhaps related to experiences of microbes in utero. So this is a study from our own group. We were really interested in whether there were the presence of microbes there was evidence for the presence of microbes in the human fetal intestine. And this was largely driven by the observation that immune maturation begins in utero. We know this, there's several studies that have shown it. And microbes that have the capacity to respond, immune cells that have the capacity to respond to microbes are evident in the fetal intestine and compartmentalized in the fetal intestine. So we examined a cohort of 50 human fetal intestinal samples, ileal samples specifically, and found evidence for a very sparse, very small bacterial burden in these samples compared to control samples. Using fluorescence in situ hybridization, we also corroborated this molecular data by observing, again, very sparse evidence for eubacterial or bacterial signal in the fetal intestinal gut. Here we're looking down on the villi, and the red stained region is essentially where we're getting a targeted staining of the presence of bacteria in these fetal samples. Using electron microscopy, we were very excited to have some of the first images of human fetal meconium in the intestine. And here we could see, and then we saw this across multiple samples, essentially pods of what look like microbial cells. They're the right shape and size for microbial cells. They're significantly smaller than any human cell type. And so this suggested to us that live microbes are present, and this is at up to 20 weeks gestation in the human fetal intestine. And so we wanted to identify what they could possibly be. So using 16S analysis, we saw that subsets of fetal meconium had evidence for lactobacillus or micrococcus enrichment compared to controls, and a large number of samples that we really didn't see a microbial signature at this stage. This is about 30% of the samples we could see evidence for the lactobacillus or micrococcus signal. Isolating the micrococcus from these fetal intestinal samples, we identified it as Micrococcus luteus. We sequenced its genome and found that it had really unique features in the genome that we did not see in other Micrococcus luteus strains that are sequenced. And these included the capacity to deal with reactive oxygen species, the machinery to metabolize hormones. And this suggested to us that these are actually evolved and selected organisms that can survive in the in utero space. And so to test this, we examined whether our fetal micrococcus isolate could actually survive in fetal antigen presenting cells, which it could compared to the control Micrococcus luteus strains, Ref1 and Ref2. The Micrococcus luteus from the fetal intestine could also grow to very low cell density on progesterone and estradiol, compared again to its control counterparts shown in the small panels above this middle plot. And really key is we showed that this strain can actually downregulate interferon gamma production by antigen presenting cells to its intracellular presence, suggesting that it's a microbe that can actually induce tolerance to its presence in the fetal intestine. So this suggests to us that our interaction with microbes begins in very early life and prenatally, and suggests that examination of maternal microbiomes and maternal exposures during pregnancy may allow us to think about opportunities for altering microbial colonization and changing the course of some of these kind of postnatal colonization trajectories that we've seen to be associated with childhood disease development. But the gut microbiome is not just related to gut associated etiologies and diseases. The data in the field suggests that it really is a fulcrum for human health. And as the data has expanded, we've seen relationships between the gut microbiome and a whole range of diseases that manifest at remote sites, including skin disorders like acne and psoriasis, neurological disorders like autism, depression, cardiovascular disease, chronic kidney disease, etc. So this is really exciting that the microbiome may actually serve in the gut, may serve as a novel target for treatment of a range of conditions that we've not considered could be treated through this route. But the key question in the field was, how do we know that the microbiome is not just a bystander and that the perturbations that we see associated with these diseases at remote sites are not just because of the effect of the disease on the microbiome? And the evidence from this comes from, for me, still a seminal finding in the field and observation from, again, Peter Turnbaugh's lab when Peter was a graduate student in Jeff Gordon's lab. And here they took stool from an obese patient and a lean control healthy participant and humanized germ-free mice. These are gnotobiotic animals that have no microbes associated with them. They're raised under sterile conditions. And what they showed was that the animals who received the gut microbiome from the patient experiencing obesity, in fact, became obese, while those who received the lean microbiome remained lean, suggesting for the first time that the microbiome was a driver of disease, of pathology in those patients. This has also subsequently been shown for kwashiorkor, a wasting disease, for food allergy more recently, and I think more excitingly for autism spectrum disorder from a wonderful study published by Sarkis Mazmanian's group in 2019, suggesting that the gut microbiome is a driver of features of pathology, not just locally in the gut, but remotely at these distinct sites. And so how might the microbiome be actually mediating its effect, the gut microbiome be mediating its effect on remote sites? Well, largely we think it's through the production of small molecules, things like short chain fatty acids, which I mentioned already, but also there's a whole range of small molecules that we're now understanding the gut microbiome produces, as you can see from this short list here, that influence various aspects of immune function. And we think of immunity as the rheostat for human health. And I'm not going to go through, in the interest of time, all of these. But what we're beginning to understand is that microbes engage in biomimicry. They can produce many of the products that we produce to regulate immunity and physiological features of the cellular features. And so this is a rich field that is ripe for a large number of discoveries, and one in which I believe we will find a large range of novel therapeutics, microbial-derived novel therapeutics. And so the concept is, how do we take this information and start thinking about translating it back to the bedside? And one of the very obvious ways is rebuilding microbiomes that are depleted of a whole range of functions that we see, for example, in very early life in children who go on to develop allergies and asthma. And the aim here is to reinstate not just microbial functions, but the metabolic productivity of these gut microbiomes that train immune function in very early life, and set up a steady state microbiome that is sustained then through later infancy and into childhood and beyond, that promotes immune tolerance. And so, one good example of this is a product that we've developed through our multiple birth cohort studies. It's a 3-member bacterial cocktail that encodes the broad range of microbial functions that are missing in infants and throughout the first year of life in those who go on to develop allergies and asthma. The concept here is that we take high risk for allergy and asthma children who are depleted of these functions, supplement them from day one, from day of birth, with this cocktail of organisms for the first year of life as tolerogenic immunity is developing and microbial metabolites shape that immune tolerance, and then determine whether this prevents allergic disease development in childhood. And so this product is now in phase 2 clinical trials with run-in short safety trials in children aged 6 years to 12 months, 12 months to 1 month, and then ultimately moving into newborns, given safety profile is acceptable in these first 2 parts. And this is actually currently almost at the end of part A1 and ready to enroll part A2, and the safety looks good. So that's using microbes as basically functional Lego bricks to reinstate functions that are missing in the microbiome. But what about precision engineering of the microbiome? And this is a very exciting field. Again, another paper from Pete Turnbaugh's lab demonstrating for the first time targeted engineering of a microbial trait in the gut microbiome. So here using bacteriophage M13 that is engineered to express a CRISPR-Cas system to specifically, in this case, excise a green fluorescent protein that's engineered into E. coli. What Peter and his group have shown is that this is possible, that they can actually go in and precision engineer and remove, in this case, a gene, a specific gene in a complex microbiome in the gastrointestinal tract. So this really opens the gates for a whole range of approaches for precision engineering of the microbiome. And then the last approach that we've been taking is really examining reconstitution at a much broader scale of the gut microbiome for a broad range of conditions that have been considered and trialed at this stage. But I think the ones that we're most excited about are the treatment, obviously, of Clostridium difficile infection, and the really landmark publication in the New England Journal of Medicine some years ago now, demonstrating that this approach, the reconstitution of the gut microbiome in C. diff infected patients, has an efficacy rate of 92%. Excitingly, similar improvements and improvements in outcomes have been observed in children with autism spectrum disorder who went through a month-long fecal microbial transplant protocol. There were significant improvements of the features of the disease in that patient population who received the treatment. And in fact, a 3-year follow-up of that population has demonstrated that not only are those improvements sustained, that they increased over time. So really exciting data coming out of that kind of gut-brain axis field. And then lastly, ulcerative colitis, an obvious target and candidate for fecal microbial transplant, given the clear relationship between gut microbiome perturbation and this disease. And so with that, I'm going to hand over to Najwa, who's going to tell you about some of the efforts that we have been engaged in here in the gastroenterology department using FMT to treat inflammatory bowel disease. Najwa?
Yeah, thank you. Can you, Alyssa, can you turn on my video? It won't let me turn it on. Okay, I can just go ahead and start talking then. I'm excited to tell you about the fecal transplant experience here at UCSF. I was fortunate to be part of the journey early on and was part of the process of getting this approved back in 2014. When we appealed to the Pharmaceuticals and Therapeutics Committee based on the need for treating this difficult patient population who had recurrent C. diff infection. Our first FMT was performed on April 16th, 2015, and since then we've done several hundred FMTs. We are able to continue this therapy under enforcement discretion from the FDA. So this is technically not an approved therapy, but the FDA has permitted its use only for recurrent C. diff. For all other indications, an IND is required in the setting of a clinical trial. Next slide. Thank you. So we've been fortunate enough to have special patient populations that we've been able to report in multi-center studies on the safety and efficacy of fecal transplant for recurrent C. difficile infection, including those with cirrhosis and solid organ transplant. Next slide. But what's near and dear to my heart and what Sue alluded to was that we're really interested in trying to understand better the role of fecal transplant in inflammatory bowel disease. And in the last few years, we've published several small trials looking at the effect of fecal transplant in IBD, specifically in Crohn's disease, in pouchitis, as well as in ulcerative colitis. Next slide. Our main focus recently has been on looking on FMT for ulcerative colitis because this is really where the data is most robust. There have been several randomized controlled trials that have been published. And what's interesting about these trials is that there's a different pearl of knowledge that comes out of each study because the methods are actually quite heterogeneous. In 2015, in the same issue of Gastroenterology, 2 studies were published side by side, but were very different. The first one was FMT delivered by nasoduodenal administration on 2 occasions. This was actually a negative study, but the study that was published from Paul Moietti's group in Ontario reported a positive outcome when FMT was delivered weekly for 6 weeks. And so looking at these 2 studies at the time in 2015, there was an interest in considering how the route of delivery may affect the clinical outcomes in patients with ulcerative colitis, especially since this is a disease that only affects the colon. But I think what's most fascinating about what we saw from Paul Moietti's study is that there was a potential for a super donor effect. So in the study, there was one donor, donor B, who seemed to have all the magic. 75% of the people who received FMT from this donor achieved the clinical outcome of achieving steroid-free clinical remission. So the talk around this time was, well, how can we figure out who these donor Bs are? Because these are who we should be using for our FMT trials. A couple years later, Param Sathi from Australia published the results of a multi-center trial where they looked at FMT again in mild to moderate ulcerative colitis, and they took a different approach. What they did this time is that they batched the donors to try to hedge their bets on what might be a donor B. So each FMT had contained material from 3 to 5 different donors. And then how they also differed is that they gave many more treatments. So in this study, FMT was delivered by colonoscopy to all the participants who were randomized to the FMT arms, and they then had maintenance therapy of one enema daily, Monday through Friday, for 8 weeks. So this was a total of 41 FMTs that was done in the study. So there was some question after this study that perhaps we should be doing a more robust regimen to achieve these good outcomes. However, a couple years later, another group from Australia, led by Sam Costello, published their results that was also positive, but this time they only gave 3 FMTs total. And the difference in this trial is that they anaerobically prepared the stool. And the thought process behind anaerobic preparation is that the beneficial bacteria that may help modulate inflammation in UC are anaerobes. So you may be delivering more of the beneficial bacteria when you're anaerobically preparing the stool. So lots of different methodologies, and hot off the presses, just published this week in the Lancet, also from Australia, from Param Sathi's group. They published the results of giving antibiotics and capsule FMT, and also achieved a positive outcome in this study. So the lesson learned from here is that maybe maybe antibiotics would be helpful, and capsule FMT may also be an option. Next slide. The question of the capsules has actually been in the field for a couple of years. Dina Gao out of the University of Alberta published the results of her trial a couple of years ago, looking at patients with recurrent C. diff infection, and found that delivery of FMT by oral capsules was non-inferior to delivery by colonoscopy. For patients with recurrent C. diff, the outcomes for cure were similar, of over 90%. Although this hasn't been studied as extensively, and certainly there's been no comparison when looking at the ulcerative colitis patients. Next slide. So looking at the data that's out there, there's still a lot of remaining questions for how we can best look at FMT and ulcerative colitis. What is the optimal route of delivery? There are several ways that we can deliver and FMT, and some are more convenient than others. How frequently should we be giving FMT? We're seeing studies ranging from 2 times to 41 times, so that still needs to be determined. What makes an ideal donor? What makes an ideal recipient? Should we be anaerobically preparing the stool? And also, should we be considering pre-treatment of patients to optimize engraftment? Next slide. And the idea behind pretreatment antibiotics and FMT comes from an ecologic principle where the intact microbial community may serve as a barrier to successful engraftment of the FMT by competition. And so disruption of the host's intrinsic abnormal microbiota may help open up the space for the FMT to engraft. And this has been shown in some mouse models as well as some small human studies as well. Next slide. So we set about to create a study where we wanted to learn more about how to optimize the delivery of FMT and engraftment of FMT in patients with ulcerative colitis. We focused on patients with mild to moderate UC, and we specifically wanted to look at the role of pretreatment antibiotics and also compare maintenance of FMT by enema versus capsule. Next slide. For the study, we included patients with mild to moderate ulcerative colitis, which we defined as a total Mayo score of 4 to 9 with an endoscopic subscore of 1 or 2. The total Mayo score is a validated scoring system that is comprised of 4 subscores, including stool frequency, rectal bleeding, findings on endoscopy, and the physician's global assessment. We did allow concurrent therapies to continue during the course of the study as long as they were on stable doses. And we also allowed patients who were on steroids to enter the study, but they had to be on no more than 10 milligrams a day of prednisone at the beginning of treatment with FMT, and we did force a wean during the course of the study. Next slide. We excluded those with severe or refractory ulcerative colitis, those with a history of colectomy, patients with disease limited to the distal rectum, and those with severe immunodeficiency. Next slide. So we had a 2x2 factorial design that led to 4 different arms, and the arms differed at the beginning where we gave pre-treatment to the patients. Next. So the pretreatment was with 3 different antibiotics. We were very broad in the antibiotic selection. So patients who received the antibiotics received vancomycin, metronidazole, and neomycin for 5 days with a 1-day washout prior to FMT. Next. Next, you can go again. And then all patients received their first FMT delivered by colonoscopy. This was also the time when we established what their Mayo score was because we obtained data regarding their endoscopic appearance. Next. Then the difference again was whether or not they were receiving maintenance therapies with FMT by capsule versus FMT by enema. For those who received FMT capsules, they received 30 capsules once a week for 6 weeks. This was observed in our clinic. And those who received FMT by enema received 60 cc's of enema administered once a week for 6 weeks in our endoscopy unit. And then all patients underwent follow-up colonoscopy. Next slide. All right, thank you. So our primary aim was to look at what happens with engraftment with the different modalities with pretreatment and maintenance therapies. And of course, we did want to look at clinical outcomes, and we defined remission as a steroid-free clinical remission at week 8, which required a total Mayo score of less than or equal to, and as well as endoscopic improvement. Next. We ended up treating 30 patients in this study, and they were roughly equal in the 4 arms. 4 patients withdrew during the course of the trial for personal reasons. It was difficult to travel back and forth, or they got a new job. 4 unfortunately had to withdraw due to the COVID pandemic because their treatment started during the just before the shutdown. So we were not able to include their results in the analysis. And then we had 2 patients that withdrew due to worsening ulcerative colitis symptoms. For these patients, we did include them in the analysis because we felt with this being a small study, they probably did represent a failure of our therapy, and we didn't want to lose their data. We had their samples included. And we ended up having 20 patients who completed their follow-up colonoscopy. For our demographics, we had 10 female and 12 male, the average age of 39, average disease duration of around 10 years. A third had been on or were on a biologic agent at the time of the study. 20% were on steroids at the entry of the study. However, I should note that many of them were on budesonide, which is a milder steroid, and half were on mesalamine. Next slide. So if you look at the clinical remission by arm, here in this graph, you can see that we found that there was a trend for antibiotic pretreatment to be superior to those without pretreatment. So 6 out of 11 who received the antibiotics pretreatment achieved remission compared to 2 out of 11 without pretreatment. Of course, this is a very small study, so it's hard to make any robust claims, but we did see this trend. We did not see any difference between those who received clinically between those who received capsules versus enema for their maintenance. Next slide. So we obtained numerous samples from our patients during the course of the study. Of course, we obtained a sample prior to the pretreatment phase, and then we obtained samples weekly during the maintenance phase, and then again 1 month and 3 months after colonoscopy. We submitted these samples to Sue Lynch's lab, where they performed the microbial analysis, and Byron Smith from Katie Pollard's lab did all the bioinformatics and put together this beautiful figure. And we compared the taxonomic compositions from inferred from the metagenomes at both the species and strain levels, and looked at the effect of antibiotics on FMT engraftment across 4 different measures. And so I'll take a moment and walk you through this slide because I really think this is like the most interesting slide that I have for you today. The 2 panels on the left represent the relative abundance of the strains from the subject on the top and from the donor on the bottom. The 2 panels on the right measure the Bray-Curtis dissimilarity, or the fraction of the community coming from the subject on the top and from the donor on the bottom. Each line represents a subject over time, and the light blue lines represent those who did not get the antibiotics, and the dark blue lines represent those who did receive the antibiotics. So looking at the top two figures, you can see that though those subjects who received the antibiotics had less relative abundance of strains from that were similar to their baseline sample and were more dissimilar to their baseline sample with the repeated FMT. And then, which, it may not be that surprising because they did receive antibiotics. However, when you look at the donor panels, you see that there is an increased amount of relative abundance from the donors in the samples from our patients, as well as more similarity and a greater fraction of their community coming from the donor. So this all suggests that antibiotic indeed enhances engraftment of FMT, and we did see that this was statistically significant. So we thought this was very interesting. Next slide. While we saw that there was a significant difference between those who received pretreatment antibiotics and those who didn't with regard to engraftment, we did not see that there was any significant difference in engraftment efficiency when looking at the different maintenance methods. So here we're looking at the similar metrics that we plotted with the pretreatment antibiotics and saw no significant difference between those who received enemas versus those who received capsules. Next slide. It's worth noting that we did, as expected in an IBD trial, have some adverse events. We had 2 patients who I mentioned before who had an ulcerative colitis flare. 4 patients in our study also reported emesis on the day that they ingested their capsules. This occurred several hours after the capsule ingestion. This was out of 7 out of 27 sessions, and all the episodes of the emesis occurred from the same donor. Also, not surprisingly, patients who were in the antibiotic arms did complain of diarrhea when they were on the antibiotics, and 1 patient developed oral thrush on antibiotics. Next slide. So in summary, this is one of the first studies of antibiotic pretreatment and comparison of enemas and capsules for maintenance dosing in the therapeutic use of FMT for ulcerative colitis. We found that there was a trend for increased remission rates after FMT with pretreatment antibiotics, and also saw that there was an increase in engraftment with antibiotic pretreatment. We have ongoing studies examining the microbial and metabolic basis of FMT response to support microbial therapeutic development, because of course we would love to see something more refined than just stool when we're trying to treat ulcerative colitis by microbial manipulation. Next slide. This is one of those kinds of studies that really takes a village. From Sue's lab to Katie Pollard's lab to my UCSF IBD colleagues, this all takes a lot of people to put together a study like this. So thank you.
Great, thank you both so much. That was fascinating, and let's bring on both of you if we can. Can we get Sue on as well? There we are. Thanks so much. So we have about 12, 13 minutes for questions. If you have questions, please type them in. Lakshmi is monitoring the Q&A. Will give me some. Let me start with a few. Sue, what would be the potential mechanism for autism?
Yeah, so again, the considerations here are metabolic mechanism. In some very earlier studies that Circus Mazmanian performed, they demonstrated ethylphenyl sulfate as a microbial-derived metabolite that when they showed it enriched in the animals that had features of ASD, and when they targeted that specific microbial metabolic capacity, they reduced the symptomology in the animals. So that's just one molecule, but it really does, again, drive us towards thinking about microbial-derived metabolites that enter the circulation and the CNS to exert their effect. I'm sure it's not the only one, but at least it gives us some inklings as to mechanisms that may underlie the gut-brain axis.
Great, thanks. What special sauce, to mix metaphors, would a super donor have to make such a person such a thing? Either one of you, I don't know. Naja, you want to start?
Well, I don't think it's really known. I think that's why people are starting to consider batching the donors. In that study from the Paul Moietti group, they did see that looking at this donor seemed to have more bacteria that produce short chain fatty acids such as butyrate that is associated with decreased inflammation in the colon. And there's also suggestion that having more secondary bile acids and bacteria that metabolize them are more likely to be beneficial. But really, like, nobody has found what the special sauce is yet. We're trying. We're looking. I'm looking for Sue to help me with this.
Yeah, and the reality is, it is a sauce. It's not going to be one thing. You're trying to rebuild a range of microbial functions into that system that's highly depleted. That's a characteristic of inflammatory bowel disease and several other conditions that originate in the gut microbiome. So it's rebuilding those functions. It's rebuilding metabolic productivity out of those organisms. And the other thing to consider here is that there was some interesting data that showed that cell-free fecal microbial transplant can also be efficacious, suggesting that the phage component of FMT may also be contributing to the efficacy of that treatment. So it's not going to be one thing, but as we start to try to define what the functional features are of an effective treatment, I think we'll get a better sense of how it actually works. And again, not thinking about one pathway, but thinking about modulating a whole range of pathways across the system.
Yeah, I'm guessing, the same way people will look back on us 20, 30 years from now and say chemotherapy seems barbaric. I'm guessing they'll see fecal pills as not being the right way. So what do you think the ultimate form factor? First of all, maybe comment on that, and maybe Najwa, you have more experience. How do you deal with the ick factor here as you propose this to people? And then second of all, sort of what do you think the end game here is in terms of the form factor for delivering this?
So I can talk about the ick factor. I've been actually so surprised how well tolerated and well received this is by patients. Anyone who's had a patient who's had recurrent C. diff knows how desperate they are. Nobody wants to do another course of antibiotics. So honestly, it's been like an easy sell. I don't even have to push it. Very often people are seeking our center because they know we offer this therapy.
And you're sitting there with a patient and you're giving them a capsule filled with stools, sort of, and you're saying, take it. You're telling me nobody ever says, oh, I can't do it.
Well, of course, they're self-selecting, right? They're coming to us because they're interested in this therapy to begin with. Okay. I actually had a lot of reservations about the dose that we were using. We were using 30 capsules at a time. All at once. And I just thought that would be hard for any pill, especially if you know what you're ingesting as feces. Mm-hmm. No one took more than a couple of minutes to get them all down. People were throwing them down handfuls at a time happily. Wow. Again, I'm sure they're self-selecting when they're coming.
Yeah, sure. And then either you or Sue, what do you think the ultimate way, form of this will be?
Yeah, I think the end game is, quality controlled mixed species microbial products to reinstitute kind of viable organisms into the system. I think it's probably not going to be in isolation. It may be that these are therapies that are administered with immunomodulating therapy as well. And then the other piece that we're really trying to push on in the field, and we need numbers to look at this as well, is kind of precision treatment of patients based on their underlying microbiology and immune status. Just as we match, organs to donors, matching microbiome therapeutics to the microbiological context that's present kind of pre-treatment, I think is really critical. But that effort requires large numbers of samples to understand what combinations and what interactions actually work and drive efficacy in the population. But that is where we're headed. And then obviously the capacity to produce those therapies, those mixed species therapies, at the GMP level for treatment of patients. And then truly understanding, developing a new matrix for matching which combination of organisms work for which underlying microbiology in the patient population.
Yeah, it struck me that now you're sort of giving you're giving a certain amount of therapy to everybody with a disease state. And so it sounds like the future world is you actually figure out whether there's an issue with their microbiome. You tailor the treatment to that, and then there's some test that you do to tell whether you've had engraftment. So you don't need 41 treatments. Maybe you've hit it on 27 or something. That's a fair
That's
statement?
exactly right. And then, real-time monitoring as well, looking for metabolic productivity out of that system, that we know that we're hitting the biomarker metabolites and actually the functional metabolites, for example, that drive efficacy in the system. So yeah, that would be the ultimate goal.
Yeah. Uh, Lakshmi tells me lots of questions on probiotics, and then another one on which antibiotics are used. So start with the easy one, which antibiotics are used, and then where do probiotics fit into all of this?
So we use neomycin, vancomycin, and metronidazole, which is a very hefty combination. But there's no right answer because this is still being investigated. But we just wanted to, if we were going to ask the question if antibiotics make a difference, we wanted to just be robust and knock out the host bacteria. So that's the cocktail that we use. And the next question was about probiotics.
Probiotics, yeah. How does that? How do you sort of think about them in the context of this discussion, and then any practical information about them?
Well, I mean, I think what we're trying to understand is what when we're talking about these cocktails that we hope to develop over time, those are essentially probiotics. But the probiotics that we have to date are so far, we don't have anything that's that effective. And the AGA recently put out a technical review looking at all the indications for GI illnesses. In adults, there's like nothing that has any strong recommendation or good data for any indication, really.
So really, all the bazillions of people taking probiotics at this moment for an adult, there's no indication where there's strong evidence it works.
No strong evidence. I mean, there's like a little bit of data in pouchitis, maybe like scant in ulcerative colitis, but really nothing to write home about. And so I don't really recommend them at this time.
And Sue, we may have saved a lot of people a lot of money. That's good. Sue, any thoughts about probiotics and how it fits
so I
into
think
your
part of the issue and what's emerged in the field is your response to a probiotic is really personalized, and again is dependent on the underlying microbiology that's there, whether those organisms can come in, whether they can actually outcompete kind of pathogenic organisms that are present in the system already. So I think how we've been testing probiotics is probably wrong. We're looking for, these products to be effective across the whole population. And I think we need to have the same approach as we were thinking about with any of these microbial therapeutics, that they have to be matched to the microbial context. The other not issue, but limitation of commercially available probiotics is they're largely the same organisms across all commercially available probiotics. It's, Lactobacillus, Streptococcus thermophilus, and Bifidobacterium, organisms that we find in very early life gut microbiomes, but not to the same extent in adult microbiomes. And, we know that the microbiome evolves over time with advancing age, and we need to think about matching the microbes to the developmental stage as well. It's another component that we need to consider here. I will say there have been some studies where some probiotics have been shown to be really effective for things like colic in babies, where Lactobacillus and Bifidobacterium play a really big role in modulating immune function at that stage of life. So I think we really have to rethink, where we are on the gradient of development, and which microbes are most active in that kind of age or developmental stage before we start thinking about developing some new therapies. So the probiotic is difficult.
Yeah. I know some people feel very strongly about it, and there's sort of there's a culture around them, no pun intended, that is interesting to think about. In terms of the little babies, and so let's assume that we find there are microbiota patterns that are associated with later asthma. So what do you think happens then? I mean, what does that end game look like?
In terms of how we treat
how you might address that. I mean, you're sort of testing a newborn for it, and then
so I mean, so mentioning the kind of the approach of trying to reinstate the functionality that's missing in those very early microbiomes, and drive a metabolic output that starts to train immune function, so that it's a reciprocating relationship between the immune response and the microbes that come into the system during that early life development. I mean, that's the approach that we're taking right now. So I
guess we'll see. Maybe last question, we're almost out of time. There, maybe for Najwa, there are questions about population differences and whether there are certain diseases within the very large spectrum of inflammatory bowel disease that you think might be more amenable or less amenable. The questioner raised the question of fistulizing Crohn's as one example.
Yeah, the data for Crohn's so far is much less promising. We actually had a pilot study a couple of years ago looking at FMT for Crohn's patients, and we actually halted it because we had a couple of people that had a flare, one of whom was bad enough to be hospitalized, and we reported these negative outcomes to the FDA and just decided to hold the study. And the trial, there's like one randomized control trial, well, nobody achieved the primary endpoint. You know, the data for Crohn's so far is not that great. There's interest in whether or not pouchitis would be, you know, a good target because it's antibiotic responsive, and actually there's data for probiotics to be helpful in recurrent pouchitis. But so far also nothing that exciting, including our own study. We saw some symptoms improve, but no change histologically or endoscopically. So really like the most promising so far is ulcerative colitis.
Great. Anything to add, Sue?
No, nothing to add on that one.
My last quick question, we're at time, but is there a difference in the delivery efficacy of coming from the top down versus coming from the bottom up? Do you lose anything going top down?
That's, you know, the question we're trying to answer. You know, looking at our study where the enemas and capsules were compared, the engraftment seems similar. So you know, we're hoping that is something that I think capsule administration seems much more feasible for our patients, especially if it's something that's going to have to be done repeatedly. So, you know, again, our study is pretty small, but it does suggest that those out would be similar regardless of delivery.
Well, thank you both for absolutely fascinating session and cutting edge session where this is really truly the state of the art, both in terms of our understanding of it and the clinical impact of it. And it does seem like there's going to be some, you know, I wondered 5 or 10 years ago, what's the ratio of hype and reality here? But it seems like there's a lot of really promising information coming out. So congratulations to both of you and your very large teams for working on this, and look forward to hearing more. Thanks so much.